Good morning, everyone, and thank you for being here today. I'm delighted to welcome you to this presentation on quality control procedures in external dosage form manufacturing and packaging. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the next few minutes, we'll explore the key aspects of ensuring quality in the manufacturing and packaging processes for external dosage forms. So, let's get started and look into how we can maintain the highest standards in these critical areas. Let's start by discussing the purpose of quality control. The main goal here is to ensure that our products are safe for use, consistent in quality, and perform reliably. We want to make sure they stay stable throughout their shelf life. Quality control also plays a crucial role in preventing issues like contamination, phase separation, or microbial growth, which could compromise the product or pose health risks. Now, another key aspect is making sure we comply with all relevant regulatory norms. This isn't just about meeting standards. It's about ensuring that our products meet legal and safety requirements across different markets. Moving on to regulations and standards, there are several important guidelines we need to follow. For instance, the FDA 21 CFR Parts 210 and 211 set the current good manufacturing practices, or CGMP, which are essential for pharmaceutical quality. Then we have the ICH guidelines, Q7, Q8, Q9, and Q10, that cover quality systems and risk management, helping us build robust processes from development through manufacturing. We also need to be aware of EMA annexes and WHO GMP standards, which are vital for international compliance, especially if we're exporting products globally. Lastly, references like USB, EP, and IP monographs provide specific quality control standards and specifications that serve as authoritative sources for ensuring our products meet the required quality benchmarks. All these regulations and standards are interconnected. They form the backbone of a comprehensive quality assurance program that supports safe, effective, and reliable products in a competitive market. Now, let's take a closer look at the core QC focus areas. First, API content and uniformity are absolutely critical because they ensure each dose contains the right amount of active ingredient, providing consistent efficacy and safety. Next, rheology, which covers the texture and flow properties, is essential for ensuring the product's stability and patient acceptability, especially for topical or oral forms. Microbial limits are another vital aspect, as they help us verify that the product is free from harmful microorganisms, safeguarding patient health. Lastly, packaging compatibility is key because it affects the stability and integrity of the product throughout its shelf life. Moving on to the overview of external dosage forms, these are the various ways we deliver our medicines outside the body, like tablets, capsules, creams, and ointments. Each form has unique challenges and QC requirements, but the common goal is ensuring quality, safety, and consistent performance. Understanding these forms helps us tailor our QC processes effectively across different product types. Now, let's look at raw material QC, which is a critical step in ensuring the quality of our pharmaceutical products. The main goal here is to validate the identity, purity, and suitability of both active pharmaceutical ingredients, or APIs, and excipients. This helps us confirm that the raw materials meet our quality standards before they go into production. When we talk about mandatory tests, the first is identity testing. We typically use techniques like FTIR, HPLC, UV, and for polymers, differential scanning calorimetry or DSC. These methods help us verify that the material is actually what we expect it to be. Next, we have assay or purity testing. Here, HPLC or UV methods are used, and we follow pharmacopoeial standards to ensure the raw materials are pure and within specified limits. Moisture content is another key parameter, especially for some materials, and we measure this using the Karl-Fischer titration. This test is crucial because excess moisture can affect stability and efficacy. Finally, for excipients, microbial limits are assessed to ensure that microbial contamination is within acceptable levels, safeguarding the safety of our product. For example, in the case of carbomer polymers, we verify their identity through infrared spectroscopy and viscosity grade before using them. This step ensures consistency and quality right from the start. Overall, these tests form the backbone of raw material quality control, helping us produce safe, effective, and reliable medicines. Let's talk about water and solvent controls. First, water quality is crucial in our formulations. We use purified water, or PW, mainly for creams and gels, because it meets the necessary purity standards for those products. For products that require even lower levels of contaminants, especially endotoxins, we use water for injection, or WFI. This ensures the highest purity when needed. Now, moving on to QC parameters. These are the tests we perform to confirm water quality. Conductivity is one of them. It helps us identify the presence of ions or impurities in the water. Then there's TOC, which stands for total organic carbon. This test measures organic contaminants that could affect product stability or safety. And of course, microbial load is critical. We need to ensure the water isn't contaminated with bacteria or fungi. For example, purified water must meet specific standards set by the USB, particularly regarding conductivity and microbial limits, before we use it in making emulsion bases. Ensuring these parameters are within limits is essential for maintaining product quality and safety throughout the manufacturing process. Let's talk about in-process controls, or IPC, which are essential for QC during manufacturing. These controls ensure that the product is being made correctly at every stage. For example, temperature control is crucial, whether we're heating or cooling the ointment base, to maintain the right consistency and quality. Similarly, we monitor mixing speed and time to ensure thorough blending, and we check the homogenization efficiency to confirm the product's uniformity. pH checks are also vital, especially during processes like cream emulsification, where maintaining the pH within a narrow range, say within plus or minus 0.2 of the target, is key to ensuring the stability of the final product. Now, IPC tests include sampling from multiple vessel points to verify content uniformity, which helps us confirm that every part of the batch is consistent. We also perform viscosity stability checks to make sure the product maintains its desired texture over time. For example, during cream emulsification, if the pH strays outside that plus or minus 0.2 range, it could affect the stability and efficacy of the product. So, these in-process controls are not just about monitoring, they're about actively ensuring the quality and stability of our formulations right from the start. Let's talk about rheological characterization. So, why is controlling rheology important? Well, it directly impacts how a product spreads, its texture, and ultimately whether patients find it acceptable to use. Plus, it serves as a good indicator of process consistency, helping us ensure each batch is uniform. Now, when it comes to common tests, there are a few key ones to keep in mind. First is viscosity, which we measure using a Brookfield viscometer. This tells us how thick or fluid the product is. Then, there's the texture profile analysis, which evaluates properties like hardness and cohesiveness, important for how the product feels during use. And for gels specifically, we look at yield stress, which indicates the stress needed to start flow. For example, in gel formulations, viscosity needs to fall within a specific range, say, between 20,000 and 40,000 centipoise at a certain RPM, to meet product specifications. This kind of control ensures the product performs as intended and maintains quality throughout its shelf life. So, understanding and managing rheological properties is a key part of product development and quality assurance. Now, let's look at the core QC requirements for assay and content uniformity. First, we need to perform an API assay, and typically this is done using HPLC or UV methods. The goal here is to ensure that the active pharmaceutical ingredient, or API, is present at the correct level. Next, we also need to verify uniform distribution across all batch containers. This is crucial because it confirms that each unit contains the right amount of API, ensuring consistency and quality throughout the batch. Moving on to the test methods, sampling is key. We take samples from the top, middle, and bottom of the bulk before filling. This approach helps us detect any potential variations within the bulk material itself. Additionally, we perform replicate analysis on these samples. This means analyzing the same sample multiple times to confirm the accuracy and reproducibility of our results. For example, when testing an ointment, we expect the API content to be within 98 to 102% of what the label claims, and the relative standard deviation, or RSD, should be less than or equal to 5%. This ensures that the content is consistent and reliable across the batch. Overall, these core requirements help us maintain high-quality standards and ensure that our products meet both regulatory and therapeutic expectations. Now, let's take a closer look at some of the key physical and chemical tests we perform to ensure product quality. First, pH measurement is crucial, especially for creams and gels, because it affects both the stability and the skin compatibility of the product. We typically aim for a pH range of about 5.5 to 7.5, but of course, this can vary depending on the specific product. Next, appearance and color check is an important visual inspection. We want to see a uniform, consistent appearance without any discoloration that might indicate spoilage or contamination. Odor inspection also plays a role. A change in smell can be a sign of chemical changes or microbial growth. Phase separation is another key factor. If the product separates into different layers, it suggests instability, which we need to address before release. For suspensions in gels or creams, particle evaluation is essential to ensure that particles are evenly distributed and not settling or aggregating. Moving on to typical specifications. For creams, the pH should generally be maintained between 5.5 and 7.5, though this can depend on the specific formulation. And, importantly, we want to make sure there's no visible separation or gritty particles, as these can compromise both the quality and the consumer experience. All these tests are vital steps in maintaining the integrity, safety, and efficacy of our products, aligning with our overall goal of delivering high-quality formulations to consumers. Let's talk about microbial and preservative effectiveness today. First, we have key microbiological controls that ensure product safety and quality. One important test is microbial limit testing, which is applied to non-sterile products according to USP 61 and 62. This helps us confirm that the levels of bacteria, yeast, and mold stay within acceptable limits. Another critical control is preservative effectiveness testing, or PET, as outlined in USP 51. This test evaluates whether our preservatives are effectively inhibiting microbial growth throughout the product's shelf life. Now, moving on to QC outcomes, we look for acceptable aerobic and yeast mold counts. This means our products shouldn't have excessive microbial presence. Additionally, we verify that preservatives remain effective over time, consistently protecting the product during its entire shelf life. For example, in the case of cream, it must inhibit the growth of challenge organisms according to USP PET criteria. This ensures that the preservative system works effectively, maintaining safety and stability from manufacturing to the end of the product's use. In summary, these microbiological controls and outcomes are vital to making sure our products are safe, effective, and reliable for consumers. Let's talk about packaging material QC. First, we look at the packaging components involved. This includes different types of tubes such as aluminum, HDPE, and laminated tubes. We also examine jars, caps, and seals, as well as box and label inserts. Each component needs to meet strict quality standards before they are used. Moving on to QC checks, the focus is on verifying the material identity and compatibility. This means ensuring that each material is what we expect and that it works well with other components and the formulation. Barrier properties are also crucial, specifically, how well these materials protect against moisture and oxygen, which can affect product stability and shelf life. Another key point is print quality and the accuracy of regulatory information. We need to make sure that labels and printings are clear, consistent, and compliant with regulations. For example, we might test a tube laminate for extractable's compatibility with the formulation inside. This helps confirm that no harmful substances migrate from the packaging into the product, ensuring safety and integrity. Overall, these QC checks are vital to ensure that every packaging component not only looks good, but also performs effectively, safeguarding the product and meeting regulatory standards. Now, let's take a look at the packaging line QC controls. On this slide, we're focusing on inline packaging controls, which are essential for maintaining product quality right on the production line. First, we verify fill volume and weight to ensure each package contains the correct amount. This is vital for consistency and customer satisfaction. Next, we check cap torque and closure tightness to make sure caps are sealed properly, preventing leaks or contamination. Label placement and barcode checks are also important. They ensure labels are accurately positioned and barcodes are scannable, which is critical for tracking and traceability. Lastly, seal integrity, including crimp and crush tests, helps confirm that seals are secure and intact, protecting the product during handling and shipping. Moving on to inspection tools, we use high-tech systems like vision inspection systems to automatically identify any visual defects or misalignments. Check weighers and torque testers provide precise measurements to verify fill weights and closure tightness, respectively. For example, in the case of tube fill weight, we typically require it to be within plus or minus 3% of the nominal weight. So, if a tube is supposed to contain 50 grams, the acceptable range would be 48.5 to 51.5 grams. This kind of control helps us maintain consistency and ensures that every product meets quality standards. Overall, these controls and tools are crucial for ensuring that each package leaving the line is of the highest quality, aligning with our overall goal of delivering safe, reliable products to consumers. In this slide, we're focusing on the finished product QC tests. These tests are a crucial step in ensuring that the final product meets all quality standards before it reaches the customer. Essentially, they serve as the final checkpoint to catch any issues that might have slipped through earlier stages. The key points here include the types of tests we perform, which can range from physical and chemical analysis to microbiological testing, depending on the product. These tests verify the product's safety, efficacy, and consistency. It's important to understand that these QC tests are not just about compliance. They are about building trust with our customers by delivering a reliable and high-quality product. Connecting this to our overall theme, rigorous testing at this stage helps us maintain our reputation and ensures that we uphold the standards we've committed to. Ultimately, these tests are the final assurance that what leaves our facility is safe, effective, and ready for the market. Let's talk about how we handle out-of-specification, or OOS, situations and the importance of CAPA in this process. When an OOS result occurs, the first step is to immediately hold the batch to prevent any further processing or distribution. Then, we need to confirm the accuracy of the test, which includes checking sampling procedures and the testing method itself to rule out any errors. Next, we perform a root cause analysis, often called RCA, to identify what caused the deviation. Once we've pinpointed the cause, we implement corrective and preventive actions, or CAPA, to address the issue and prevent recurrence. Now, let's look at some typical causes of OOS results. Common issues include inadequate homogenization, which means the sample wasn't mixed properly. Deviations in pH or viscosity can also lead to OOS outcomes, as these parameters are critical to product quality. Additionally, preservative failure might be a culprit, indicating that the preservative didn't perform as expected. To put this into perspective, consider an example where we encounter an OOS viscosity result. The investigation might reveal that the mixer RPM settings were not optimal. In response, we would retrain operators on proper mixing procedures and update the Standard Operating Procedure, or SOP, to ensure consistency moving forward. This process is vital because it helps us maintain product quality and compliance, ultimately supporting our overall manufacturing excellence. Let's take a closer look at documentation and best practices, which are crucial for maintaining quality and compliance. First, under documentation essentials, we have SOPs and validated methods. These are our standardized procedures and validated techniques that ensure consistency across all operations. Then, there are batch manufacturing records, which provide a detailed history of each production batch, offering traceability and accountability. QC test reports and certificates of analysis are vital for verifying product quality and meeting specifications. Lastly, deviations, out-of-specification results, and CAPA logs are essential for capturing issues, investigating root causes, and implementing corrective actions to prevent recurrence. Moving on to best QC practices, systematic training and competency checks are key to ensuring our personnel are always equipped with the right knowledge and skills. Risk-based control, as outlined in ICH Q9, helps us focus our resources on the most critical areas, enhancing overall quality. Routine calibration and equipment qualification are necessary to maintain accuracy and reliability of our instruments. Finally, monitoring trend and stability data is a proactive approach to detect potential issues early, ensuring our products remain safe, effective, and of high quality. These practices, when integrated into our daily routines, support a robust quality system that aligns with our overall goal of delivering safe and effective products. For further support, please feel free to reach out to us. We're here to help you with any questions or additional information you might need. Remember, you're not alone in this journey. If you need guidance or clarification, don't hesitate to contact us. Thank you so much for your time and attention today. We truly appreciate your engagement and interest. And again, thank you for being part of this presentation.